Welcome to our second week of Biotech 201 after our snowy interruption. It does not seem like a week ago we had just made it through one snowstorm and we're now awaiting the second one. It feels like springtime outside, doesn't it? It's incredible. Thank you for your patience as we worked through all that. I hated having to cancel, but I just felt that it was better and safer if we didn't have anybody out on the road. So we will run one more week. We'll run into the 4th of March. And I know that for many of you, you may not be able to be here on that last session because you are away or have other plans. If you'll see Stacy Brewer out in the atrium sometime today or next week, we'll give you access to the online version. So if you're out of town, you'll at least be able to stream online. And those of you that are watching from our online audience, it's great to have you with us tonight as well. And remember, we have the chat session going and it'll be moderated, so please feel free to ask questions as we move on. So before we dive in, the same caveats that I told you 2 weeks ago are still in force. Remember, correlation does not equal causation. These are new studies. In many cases, I'm going to show you one study that has this suggestion. That doesn't mean that you need to rush out and do something. Don't necessarily try this at home. So recognize those caveats are still in play, and they're especially more so in play this week and next week as we talk about new findings that link the microbiome to specific disease. So let me actually show you where we're going to go tonight. We're going to talk about the microbiome of our home and the bacteria that are present in your home. Again, do not stop at the grocery store on the way home and buy antibacterial everything in the world. Please do not do that. And then we're going to talk a little bit about the microbes that live on our skin and if those microbes seem to have any relationship to the preference for mosquitoes nibbling up on our skin or nibbling on our ears. And then we're going to transition into focusing on our gut microbiome. And we're going to talk about how potentially alterations in our gut microbiome may influence behavior. We'll talk about autism, and we'll talk about cardiovascular disease. So we have a lot of ground to cover, a lot of details for us to dive into. But before we start on new stuff, an observation and then a response to a question that I was asked. This is really important. Most of the microbiome studies that have been done to date are weird. In other words, they have primarily been performed in Western, educated, industrialized, rich, democratic nations. There's nothing wrong with that at all, but recognize that that is potentially a very skewed set of bacterial populations compared to the rest of the world. So there are more and more microbiome studies being done looking at other populations, looking at hunter-gatherer populations, and populations that don't live under the same conditions that we're in at this moment. It'll be really interesting to see what those find, because those may, in fact, be more like what our ancestral microbiome was than the microbiome of the current 21st century. So stay tuned for that. And then the second piece, last week, we talked about how a newborn, 2 weeks ago, we talked about how a newborn's microbiome is colonized at birth with bacteria from the birth canal or, in case of cesarean section, from the skin of the newborn's caregivers. And I had this fantastic question that was asked of me at the end, as everyone was on their way out the door. How does the immune system tolerate all these bacteria colonizing our bodies? If our immune system is so good at detecting foreign invaders and detecting bad bacteria, how does it allow all of these millions of bacteria to set up shop, essentially? So I did a little bit of digging, and there's a paper that came out that's in mice. So let me make that really important qualification, because mice and humans are not the same thing. You'll hear me say that a couple times tonight. But in mice, they identified a specific type of white blood cell that is present at very high percentages very early in life. In fact, it's almost never seen in adult mice. But in the first 2 or 3 weeks of a mouse's life, which corresponds to the first many, many months of a human's life, even maybe the first year or so, this specific population of white blood cells seems to suppress the immune system. And then as the mice enter the second week of life, the percentage of these white blood cells drops by 50 or 60%. And then by the third week of life, these white blood cells, these suppressor white blood cells, are almost non-existent. So the thinking, or at least the hypothesis, may be that let me back up. As these white blood cells drop, the newborn mice are able to actually detect and destroy infection. So their immune system is allowed to ramp back up. So the thinking may be that at least in mice, this suppression allows the microbiome to colonize and to take root. I haven't seen anything yet that says if we have something similar or not, so we'll stay tuned. But potentially we have some built-in system that says, hey, we need to be careful. We need to not immediately attack everything so that we can allow the microbiome to form. And many newborns are susceptible, especially preemies, are susceptible to infection even more so than you might expect, which potentially could be because they have an intentionally suppressed immune system. All right, so now let's jump into this week's content. Let's start with the microbiomes of your home, or the microbiome of what are called built environments. In other words, man-made spaces. Every built environment quickly acquires its own microbiome. And much like the microbiome of infants, that microbiome is determined by the people that come in contact with that space. So I'm going to show you two studies, and they both have small numbers, and I recognize that. Again, this is early going. This is a study, this paper was published in this past year, The Diversity and Distribution of Bacteria in Residential Kitchens. And this looked at 4 homes in Boulder, Colorado, and they looked at 82 different surfaces in those homes, from countertops to the floor underneath the refrigerator to the water faucet to the touchpad on the microwave oven. Now, this image is showing you many, but not all of those 82 surfaces. And you'll see here that the y-axis is the average number of OTUs observed. These are taxonomic units. This is a measure of the number of bacterial types that are present. So it isn't necessarily the number of bacteria present, it's the number of different groups of bacteria that these researchers were able to classify. So there are thousands and millions of individual bacteria for every single type, but this is a measure of the richness of the diversity of the bacteria that are present. And you can see we've got some things down here that don't have a whole lot of bacteria present, and we have some things up here at this end, at the right end of the chart, that have a lot of bacteria present. So let's explore that just a little bit, and I know you're going to probably mentally be thinking about your own kitchen as well. Again, resist the urge to run home and wipe everything down. So at the left end of this bar graph, these are the relatively less diverse populations in your kitchen. Your kitchen faucet has a relatively small number of different types of bacteria, as does, surprisingly enough, your garbage can. Potentially because you may wipe down your garbage can relatively frequently. Many of you may wipe down your garbage can frequently. In the middle are the surfaces that we tend to touch a lot. The handle of the refrigerator, the handle of the oven, the stovetop. More types of bacteria, higher diversity of bacteria. A little bit higher, the bottom shelf of the refrigerator that collects everything. And the seal around the freezer. I don't know my wife's a great homemaker and housekeeper, and she may clean the seal of the freezer, but when it's my turn to clean, there is no freezer seal and cleaning happening in our house. And then the highest frequencies, the floor underneath the sink, and the exterior of the stove fan exhaust. That potentially not so surprising because you are sucking up large amounts of air and any bacteria that may be present in that air, and they stick to the surface of the exhaust fan. And that probably gets cleaned very, very rarely. But so that gives you a sense of just general places in your kitchen and the diversity, the bacterial diversity. Now this group has done some really nice visuals with this. They've taken just a standard sink, a standard kitchen layout, and they've color-coded all these different places. And they were able to identify, based on the type of bacteria they found, if those bacteria likely came from contact from human skin because we touched it like cabinet drawers, or if they are the bacteria you often would find on produce, vegetables, and fruit that you might bring in either from the grocery store or the garden, or if they're bacteria that are often found in water. So they might have come out of the faucet and then maybe splattered or aerosolized in the sink and landed on other surfaces. And so each of these three different images shows those different sources. On the left-hand side, bacteria from skin source, the middle side is from produce, and the far right from water. And the depth of color, the darker the space, the greater the concentration of bacteria. So if we start all the way on the right-hand side, the faucet, letter C, pretty much the only thing that's dark blue is the faucet itself. So that had the highest concentration of waterborne bacteria in the entire kitchen. And then if you look at produce, there's a fair amount on the countertops or on the stovetop. That's where the color is a little bit darker. And on the skin, the darkest colors are on the handles of the cabinets, which is not surprising, and on the entire faceplate and the handle of the drawers, also not surprising, and places like the garbage can, places where the skin comes in contact, the handle and the door of the refrigerator as well. So you can get a sense of where the bacteria live in your kitchen. They also found, although at very low levels, the bacteria that you do not want in your kitchen. The Salmonella, the E. coli that can be harmful. Remember, the vast, vast majority of our bacteria are helpful or just benign. But there's a small percentage of bacteria that you really don't want in your system. And again, you can see by these percentages, these are really low overall percentages in the overall bacterial population. And they found them in certain places. For example, they find a relatively high frequency of Salmonella on the touchpads of the microwave, potentially from handling chicken or ground beef, putting it in the microwave to defrost, and then touching the touchpad. So that's one study. Now let me show you a different study. That was Boulder, Colorado. This is a study from Raleigh-Durham, North Carolina. They looked at 40 houses. They looked at a smaller number of overall locations, but looked at locations throughout the house, not just in the kitchen. The cutting board, the kitchen counter, the refrigerator, the commode seat, a pillowcase, a door handle, TV screen, and then the upper trim around the door inside, and the upper trim around the door that's an exterior door as well. And you would expect that the bacteria on surfaces like the television and the interior and exterior door trim are not things that you will have put there by human contact, but they're things that likely will have floated to that space and landed and taken up residence. This is a box and whisker plot, this image, this graph that you see. The dark line in the center of the box is the average number of these taxonic units. The average number, it's a measure of the diversity. The box that surrounds it is a measure of the 95% confidence intervals. It's a measure, a couple of different things, without going too deep into statistics. It's a measure that says, I am 95% certain that the real value, the real average in my population is within these bounds. So this is based on the bar is based on the 40 or so samples. This is, what's the likelihood that my population average is somewhere in here? That's not quite an ideal way to explain that, but I think that works enough. And then the hashed bars represent, in this specific image, they represent the minimum and the maximum amount of units of bacterial types that they found among those samples, with a few exceptions, and those circles are the outliers, so we're not going to worry too much about the outliers. The main take-home point, that's enough for statistics and for interpreting graphs. The main take-home point is that there's a pretty broad range of variation in the diversity of bacteria that you find in these surfaces. These first three, the cutting board, kitchen counter, and refrigerator, being surfaces that are likely to come in contact with food. The next three, the commode seat, the pillowcase, and the door handle, being surfaces that are likely to come in contact with human skin. And then the last three being the ones where it really just is floating through the air or aerosolizing. Now, I showed you an image like this. This is another principal components analysis, and I'm going to have a conversation with David later this week so that I can actually tell you even more about how principal component analysis is put together. But the key point here is that the closer two dots are, the more similar they are in the type of bacteria populations present in those spaces. So you'll notice this first circle that covers this kind of periwinkle blue. These are the types of bacteria from the 40 houses that are found on the outside door frame. And you can see that they are physically in a different part of this graph than most of the rest of the bacteria. These are the bacteria that primarily are leaves and outdoor types of bacteria. That's what you'd expect. This is on an outside door. The rest of the bacteria are a little more closely aligned. I don't know if I should show you this or not, that the bacteria from the pillowcase in your toilet seat are very, very similar to each other. In part because they both come in contact with your cheeks. Yeah, I know, that was a cheap laugh. I know. But they both come in contact with human skin, so you would expect that they're going to harbor the bacteria that are found primarily on human skin. So it's interesting to get a sense of this kind of information. What does it tell you about the bacteria that colonize your house? So the authors of this study essentially put this quote in that I want us to focus on. So the previous part is, this work provides further evidence that skin-associated bacteria are commonly dispersed throughout the built environment. So in other words, that in something that we create, that we then inhabit, there's a lot of bacteria that are found from skin. And that they can survive for extended periods of time, and that they're obvious, and this makes sense, they're more common on touched surfaces. So they talk about the fact that our bodies are where most of these bacteria come from, and our homes likely carry an identifiable signature of our own microbiome. So I could go into your home and gather bacteria, and that would give me a pretty good sense of the type of bacteria that are likely found on your own skin. Your home has now been colonized to match you. Now, if you're moving into another home, over time, the bacteria of the people that lived there, or the dog that lived there, or whatever, the cat, will ultimately trade out and be recolonized by your own bacteria. And we aren't even going to think about spaces where there are lots of people moving in and out like hotels. But there's lots of skin, and again, not harmful bacteria, not bad bacteria. So before I get myself in trouble with the tourism industry, let me tell you this. This is not in your handouts. The healthy human produces a convective plume. So that means it's like continually rising up above and around your body of 37 million bacteria per minute. You are shedding 37 million bacteria per minute just out into the atmosphere. So when I saw this quote, it reminds me of Pigpen. Now, Pigpen says that he carries with him the dust from ancient civilizations. There's a lot of bacteria in that dust from those ancient civilizations, but he's also producing a whole lot of cloud of his own bacterial microbiome as well. So what can we learn about skin bacteria microbiome? So that transitions us into the next piece that I want to talk about just really briefly. I just want to give you, I'm doing a lot of conversation about your gut microbiome. But I want you to realize that the microbiome of other parts of your body also are really interesting and have potential insight. So we're going to spend just a little bit more time in the skin before we dive to the gut. So this is looking at the influence of the bacteria on your skin and how attractive you are to mosquitoes, specifically the type of mosquitoes that carry malaria. Now, some of you are, show of hands, those of you who mosquitoes are always, who love you, the mosquitoes are always around you. Okay, those of you who the mosquitoes just seem to leave alone? Okay, so there are lots of cues that mosquitoes use to identify a host and to land and locate on it. Heat, moisture, the vapor from your breath, visual cues, but chemical cues play a really large role in helping the mosquito land and identify you as their next meal. And some of these chemical cues are driven by the bacteria that are on your skin. Human sweat has no odor. The bacteria on your skin use that sweat, take it in, metabolize it, and some of the byproducts they produce have an odor associated with them. They're aromatic molecules. And so it's the bacteria on your skin that give you that distinctive aroma, not the sweat itself. And so the thinking is that potentially the specific bacteria on your skin that take the sweat and metabolize it into aromatic molecules may play a role in helping mosquitoes know if they want to find you or if they're just going to leave you alone. It's an interesting finding, interesting theory. So this paper was published in 2011. Attractiveness. So they took 48 men and they measured how attractive these men are to mosquitoes. Now, they did that in a couple different ways. No men were put in a box with, this is not like the commercial for the, for this anti-mosquito product is better than this one. But there was a way that they measured the attractiveness, and they ranked these men, and there were some men that were highly attractive to mosquitoes, and some men that were poorly attractive to mosquitoes. I wonder if they told these men, I'm sorry, but you are poorly attractive to mosquitoes. So this is what they then did. They looked at the highly attractive and the poorly attractive men, and they specifically looked at the skin on the bottom sole of the foot. I'm not 100% sure why they did that. Apparently, those bacteria seem to play a large role in producing the volatile aromatic chemicals. And we'll just leave that at that as well. But then what they did was they took these men and they looked at the microbiome of the foot, the skin on their feet. And they found that the highly attractive individuals had less of a diverse population, so there were fewer different types of bacteria, and specifically there were more in number for Staphylococcus. And contrast that with the poorly attractive men, which had higher levels of Pseudomonas, higher numbers of Pseudomonas bacteria, and an overall greater diversity in the different types of bacteria. Now, this was at 2011. This was really early in the process. But what I find intriguing is the statement that the authors make in their conclusion, which is identification of the volatiles, so the aromatic molecules produced by these genera, so these specific bacteria, may lead to the development of new mosquito attractants or repellents. So in other words, if you can figure out what bacteria are on the skin that attract mosquitoes, then maybe you can figure out how to make those compounds synthetically and put them in mosquito traps to draw mosquitoes towards them. And if you can figure out what bacteria are on the skin of the poorly attractive men and figure out what kind of chemicals they metabolize out of the human sweat, then maybe you can actually make mosquito repellent from that. So it'll be interesting to see what comes down the pike from this. But another interesting way that the bacteria in and on your body impact the world around you. So the next time you're in the presence of someone who says the bacteria just bite me all the time, and someone says that's because you're so sweet, because in the South that's the kind of way we say things, you can actually say, no, actually it's the bacteria on your feet that produce the things that really make yourself smell good to the mosquitoes. And then you will not get invited back to that party. All right, I'm going to shift gears on us now. And there's an emerging concept, and it will be really interesting 3 years down the road, 4 years down the road, if we do when we circle back to this topic for Biotech 201, to see if this really bears fruit or if it was just an early idea that didn't pan out. But the thinking, historically, we've said that disease was caused by either the genes, changes in the DNA inside your cells, or by some environmental insult. Maybe something that you were exposed to, maybe an infection, maybe your diet. And in many cases, it's the combination of the two that played a role. We're adding a third category, which has both genetic and environmental pieces to it, but that some component of many diseases may actually be determined by the bacteria in and on your body, and by overall changes in the population of those bacteria. So if you suddenly lose a subset of bacteria or you gain another set of bacteria you didn't traditionally have, that may throw your body off balance in a way that leads to symptoms of disease. And I'm going to walk us through a few of those issues tonight, and we'll dig into more of those issues tomorrow. Let me stop and see if we have questions, either from those of you in the room or from those of us on our online audience. Bob, and then I'll come right up here.
My hands spend all day touching things that aren't me.
Your hands spend all day touching things that aren't you, yes.
So unless I suck my thumb a lot, I would not expect my gut bio to match my skin bio. Is there any correlation with that, or is that relevant?
Yeah, so your hands touch lots of different things, but most of the time we don't knowingly put them to our mouth. So is there a correlation between the microbiome of our gut and of our skin? Early in life with those infants who are born by cesarean section, there's very much a correlation. Later on in life, many of those things that you ingest on your hands, your immune system recognizes and attacks. But there certainly are things, we scratch our nose, or we look in the mirror and we pick our teeth, or we wash our face, and so some of those things get transferred into our digestive system. And if our immune system can't catch them and they take root, then yes, you can end up with real challenges. A lot of times, so if you go to a restaurant and you have a sandwich, the bacteria that are on your hands are transferred to the bread, which then is transferred into your digestive system. So the transfer can occur in other ways as well. That's a great question.
What about people, like you're talking about, when somebody moves into a different home, eventually the environment is going to change. What about people that begin to live together and then over a long period of time, does their microbiome?
So do the microbiomes of people that live together mimic each other? There is some evidence that says the answer to that is yes, that there are similarities in the internal microbiome of people that share the same environment. And that's not terribly surprising. I'd love to see a study that looks at newlyweds and looks at their microbiome before and then during the first several months to see what sort of changes. I haven't seen anything like that. Doesn't mean it's not out there, but yes, you would think the results I've seen say that yes, that is a possibility.
He might become the mosquito magnet instead of you.
Oh, he might become the mosquito magnet instead of you. I don't know, because I don't know how your microbiome in many ways is now I'm going to say something that sounds like the total opposite. Your microbiome is relatively stable throughout life. It's possible that you're going to see changes, but they're going to be a little more subtle as opposed to major shifts.
You talked about if there's a temporary or transient change in your microbiome. Is there an implication there about things like foreign travel? There's always the classic case of somebody miles in his revenge and all this wonderful stuff. Is there possibly a deeper thing than that would be more environmental? And if that's the case,
So if
what should you do
if you travel to foreign countries, can you pick up the bacteria from those surfaces and bring that in, and can that bring about a shift? A temporary shift, or maybe not a temporary shift. The answer seems to be yes, it's possible. How often it occurs is really going to depend on what you are exposed to and how often, and your own microbiome health. If your microbiome is in a state of transition or a state of flux, it is going to be much easier for another species to find a niche to take up residence, as opposed to if you have a stable microbiome. Yes, sir?
Good example that might be when you go to a country that has not a good water supply. And you drink the water, you get sick. The native population drink it, they aren't getting sick. You're used to the new microbiome that you're not adapted to.
Yeah, so I think that, that is a great example of that. Especially if you have taken antibiotics, for example, before you go, and you drink in a water source that contains bacteria that are not the type that have colonized your gut, that can be really problematic. I spent some time in India, and I know that everywhere I went, it was drink-only bottled water. No ice, no nothing, because I was not used to the microbiome that I would have found had I drunk the water.
What percent would you inherit from your parents or of your microbiome?
What percent would you inherit from your parents? So I'm going to take the word inherit out of the way. What percent might you acquire because you're in the same space as your parents? An infant's early microbiome is going to be very much dependent upon the mother's microbiome, especially as they pass through the birth canal. But for example, if you have an individual who's adopted and is adopted at birth into another family, their microbiome, aside from what they take in at birth, their microbiome is going to come to look more like the people that they live with than the people that they share genetic information with. Any others? Let's take one more. Yes, ma'am.
A long time ago, I recall reading in Scientific American that it was the blood type that mosquitoes like, and the type O was the type they like best of all.
That they like type O. Mosquitoes like that the blood type plays a role in that.
Is there any validity there?
I'm going to have to plead ignorance to that, although I'm just going to have to say I don't know before I step foot in something that I have no business even trying to talk about. I will try to find that out. That's a really good question. That's a good question, because I would imagine there is going to be very difficult for them to sense your blood type without seeing your blood. Now, it may be that has to do with if once they pierce your skin, how long do they stay? I don't know. I'll try to find that out. One more. Push that envelope. Yes, sir.
Can you describe
The microbiome?
Yeah. Can you, is that plant, animal?
It is bacteria. It's bacteria, virus, and fungus. So your microbiome are bacterial, viral, and fungal inhabitants. Most, but not all of them, single cell.
They're not mobile either.
They're not mobile. Well, no, that's not necessarily true, because some of them actually have cilia or flagella that allow them to navigate through. Okay, let me move us on. Excellent questions, as always. So let's talk about your gut microbiome. So your gut microbiome, we talked about this last week some, carries out several functions. It plays a role in your immune response and priming your immune response. This was the old friends hypothesis we talked about 2 weeks ago. It also plays a role in helping your gut move food and things through it in a timely manner. It plays a role in helping absorb nutrients from the food and passing that through the body. Next week we're going to talk specifically about how some microbes in your gut do a really good job at breaking down food that normally your cells wouldn't be able to digest and provide you with extra calories. So we'll dig into that when we talk about obesity and diabetes. But there are multiple functions of the bacteria in your gut. And remember, we showed this image at the top of all of these small villi, these tiny finger-like projections that line your digestive system, and that there are bacteria that are in a mucus layer right over top of those, and that those bacteria interact both with the cells of the villi, your cells, and with the environment of what's flowing through the gut system. So in the last few years, this concept of a link between the microbes in your gut and your brain has really begun to arise. And some of that's due to animal studies. For example, we know that if you expose animals to high levels of stress and you put them in an emotionally stressed state, the populations of the bacteria in their gut change. But we're beginning to see evidence that this is a bidirectional relationship, that the specific bacteria in your gut also influence the way that your brain senses and responds to things. And that happens through a number of different ways, but one of those is, actually before I talk about that, let me talk about how we actually are going to regulate, how we're going to test that. You've heard me say a lot that correlation is not causation. We find lots of times where we see differences in the bacterial population of two individuals with and without disease. But we need really rigorous approaches where we can very intentionally alter the bacterial population and then study to see if there are any differences. So for the next 2 weeks, you're going to hear me talk about almost all of these types of experiments. In this specific instance, experiments with mice, but many of these also extend to humans. So a lot of work is done at about 9 o'clock on this chart. For those of you that are watching online, the germ-free studies. These are mice that are born in completely sterile conditions, and they have no microbiome. They don't get any microbiome from birth because they're often delivered by C-section. Their food is sterilized, their air supply, their water supply is sterilized. They have no bacteria in their gut or on their skin or anywhere. These mice represent an incredible research opportunity because you can very specifically create a microbiome for these germ-free mice with your chosen set of bacteria. So you can build your own microbiome for these germ-free mice and study them. Right below that are infection studies. You can also take these germ-free mice and you can expose them to specific bacteria to see how the body responds to it. And does it respond to it friendly or as a foe? This one at the bottom, fecal transplantation studies. We're not going to talk much about that this week. We're going to spend a lot of time next week talking about poop. But this essentially is taking the bacteria that live in the gut of humans or of other mice and transplanting it into these mice to see if changing the microbial population or building a microbial population influences a certain set of symptoms. Antibiotic and probiotic studies are also used a lot. Antibiotics generally wipe out bacteria. Some antibiotics wipe out certain types of bacteria, rod-shaped bacteria, gram-positive, gram-negative. Other antibiotics are broad spectrum. They wipe out lots of different bacteria. So you can use them if you want to suppress the micropopulation in an individual or an animal's gut. In the alternate way, you can provide them with probiotics, which remember are specific strains of bacteria that may or may not be helpful, and you can supplement the animal's microbiome in a very specific way. So all of these pieces become part of how we begin to understand the role of the microbiome, not just for brain, but also for any other disorder. So you're going to hear us talk about these different pieces, and I wanted you to kind of have that background of the research story. All right, so let me give you an example. So this is Walkerton, Canada, and in 2000, there were very, very heavy rains that washed agricultural runoff into the water supply that served the municipality of Walkerton. And there was a specific strain of E. coli that got into the water supply, and over half of the town of Walkerton got sick with enormous gastrointestinal distress, and 7 people died from it. So there were a lot of studies that have followed up in the 14 years since the Walkerton incident, looking at the long-term impact of dramatic changes in an individual's microbiome from this E. coli infection. In 2002, researchers began asking residents who had been exposed and who had gotten sick from the E. coli, asking them questions about other parts of their health. And they discovered an unusually large number of individuals reported depression or post-traumatic stress disorder or high levels of anxiety, much more than they would have expected from the general population and the population dynamics. Now, it's very possible that having a major long-term illness like an E. coli infection, just that in and of itself could cause anxiety, depression, and other symptoms, having nothing at all to do with the specific microbiome piece. But the researchers wondered if potentially some of these behavioral differences were because the gut microbiome had really changed in these individuals. And it wasn't simply that they had been exposed to some long-term illness, it was specifically that their microbiome had been altered. If you were in Biotech 201, wow, like 4 years ago when we talked about neurodegenerative disorders and we talked about psychiatric disorders, we talked about neurotransmitters. Neurotransmitters are tiny chemicals that are produced primarily but not exclusively in your neurons. So in your nerves and in your brain cells. And these neurotransmitters are important signals for how your neurons communicate with each other. So when two neurons are next to each other, there's a little gap between them, and the neuron that's receiving the first signal that wants to pass it on to the next signal releases these tiny chemicals, these neurotransmitters, into the space between the neurons, and they travel across the space and they interact with the neuron on the opposite end, and it carries a signal forward. It's a way of communicating. There are lots of different kinds of neurotransmitters. Some of them control movement, some of them control mood, some of them activate, some of them suppress. I was really amazed to discover that bacteria are capable of producing neurotransmitters. A lot of neurotransmitters. So you can see on this slide the different types of bacteria that produce the specific neurotransmitters. So Escherichia and Bacillus and Saccharomyces produce norepinephrine. Norepinephrine and serotonin, these first two, these are the major neurotransmitters that anti-anxiety medicines act upon. These both play a major role in the anxious and the fear response. Dopamine plays a major role in movement, also plays a role in depression. Gamma aminobutyric acid, also called GABA. So the bacteria in your body are capable of producing neurotransmitters. Well, okay, so they're in my body, they're not near my nerves. Well, no, actually these neurotransmitters can actually cross across the cells in your intestine and make it into your bloodstream and travel to your brain. Or they can travel to the nerves that innervate into your gut. So it's at least theoretically possible that if these bacteria are producing certain neurotransmitters and you alter the population and you alter the amount of neurotransmitters, that may lead to changes in the neurochemical balance in your brain and your mood. A really interesting theory. So a lot of groups have begun doing some tests on this with animals. So I'm going to show you some work in mice. So this is measuring anxiety in mice. Okay, I know you cannot just ask a mouse, are you feeling anxious, are you worried? That is a major challenge with this study, so I recognize that. You do this by studying their behavior. You put them in situations where you record how well do they respond to situations that might be fear-inducing. So one of these is you put them in a maze, and the maze has some open arms, and one of the arms is well lit, and one of the arms may not be as well lit, and you look to see where does the mouse go? Or you look to see, you put the mouse in a big box, and you see, does the mouse spend most of its time around the corners, kind of in protection, or out in the open spaces? And mice that generally have less anxiety are in the open areas, the open arms of the maze or the open spaces of the box more. You also can measure depression in mice. And one way that you measure depression in mice is that you have the mice swim, and the mice swim for extended periods of time. The mice don't drown. The mice swim for extended periods of time, and if the mice are depressed, they are immobile and they just lay in the water for long periods of time. So you measure the amount of frozen mouse time as a marker of depression. So we have normal mice, and we're measuring anxiety. You know, isn't it amazing the kind of research project? I mean, it's amazing. It's actually, but what is even more amazing is that this correlates well with behavior. I mean, this is not just somebody randomly saying, let's put the mouse in a tank and watch them swim. There is enough years of good evidence behind this that the scientific community accepts these as markers of measurement of mouse behavior. Yeah. So here's what's interesting about these, and we'll stop for our break in just a second. They are comparing mice that were fed just a simple broth to mice that were fed the broth that was supplemented with a specific lactobacillus, Lactobacillus rhamnosus. This is a lactobacillus probiotic that you can go to the grocery store and you can buy. So they gave this probiotic to these mice, and then they put them in the maze and they put them in the water test, and they measured their levels of anxiety. The bacteria-fed mice showed lower anxiety, and when they looked at the level of corticosteroid, a stress-related hormone, these mice had lower levels of the stress hormone. So let me show you this image. So moving from left to right, the solid black box is the mice that were not under stressful conditions that were fed the broth. The gray box are the mice that were stressed and were fed the broth, and you can see there's a higher amount of the stress hormone produced. The mice that were not stressed and were fed the probiotic, they don't have a whole lot of the stress hormone. That's not surprising. But the mice that were stressed and fed the probiotic have a significantly, in the striped box here on the right, they have a significantly lower level of stress hormone. That's the only difference between these mice. They're inbred. These mice genetically are the same. They're raised in the same environment. They're put in the same test. The only difference is if they get this probiotic or not. And then if you look at so this is the elevated maze test. This measures anxiety. The higher the bar, the more time they spend out in the places that are open, so the less anxious they are. So the mice that were given the probiotic, in this case, those are the white boxes, spend more time in the open arms, less anxiety. This next one, the swim test, this measures the amount of frozen time. The solid black box is the mice that were put in the swimming test and just given broth. They spent more time frozen immobile than the mice that were given the probiotic. So at least in this study, and again, do not rush to the store. Do not walk through the aisles of your grocery store handing out samples of Lactobacillus rhamnosus. This is one test, and it's a test in mice. But it's intriguing. It has a connection. And here's the other really interesting thing. There's a nerve, the vagal nerve, that plays a role in connecting with your digestive system. And if you impair that nerve from functioning in these mice, then the mice that are given the probiotic don't show a reduction in anxiety. They don't show a reduction in depression. So this is clearly interacting through that specific biological nerve interaction system. All right, one more slide, then we'll take our break. So how about in humans? So for individuals that have major depressive disorder, many of them, up to a third of them, do not respond to the typical antidepressive medicines. And a small study has actually found that if you administer an antibiotic, minocycline, that if you administer this antibiotic, they respond and the depression lifts. Yeah. Now, there are other studies that have said that individuals actually get worse with some of these medicines. So again, please do not suddenly go out and this is a small study. I'm showing you this to say this could be. Individuals with chronic fatigue syndrome, a sub-study of them, of these individuals, were given a different lactobacillus, and they experienced a reduction in anxiety. And then this other study, and I'll spend a little bit of time on this, these were volunteers that were given two different probiotics, a lactobacillus and a bifidobacteria. They were given this for 30 days, and they were tracked with their psychological distress, and then they also measured levels of cortisol, so stress, a stress marker. What's important about this study is that it was double-blind, placebo-controlled, randomized. And many of these studies aren't. This means that neither the people taking the supplement nor the people that were assessing them knew who got the supplement and who got the placebo. And it was randomized as to who got the supplement and who got the placebo. So this is the kind of study that you want, as opposed to, I asked these people to take this probiotic, and I asked these people to eat rice, and then I asked them about their own health. Well, yeah, I know I'm taking the probiotic. I'm going to look, it's the placebo effect. I'm going to look and expect to see something different. This is not that kind of study. All right, here's your teaser. When we come back, we're going to talk about autism and your microbiome and some very early initial studies with some pretty intriguing evidence. We're going to take 10 minutes, so it is 7:20. We're going to come right back here at 7:30. Answer the question about mosquitoes and blood type. So where was the question? All right, right here. Okay. So I should wait until she actually makes it back in the room, right? Okay. So the fantastic moderators that are watching the online session and responding to questions heard the question about, do they prefer blood type? Or do mosquitoes prefer a certain type of blood type? Did some Google searching and sent me some information back. And one specific study found that in a controlled setting, mosquitoes landed on people with type O blood nearly twice as often as those with type A. And people with type B fell somewhere in the middle. So apparently, based on other genes, about 85% of people secrete a chemical signal through their skin which indicates which blood type they have. So there we go. So there is, and that they don't have to taste it to know. They're based on other chemical secretions. All right, let's jump to autism. Now, let me say this at the very beginning. From what we understand about autism, there are multiple different pathways that lead to autism. Multiple different genetic changes, multiple different environmental factors, and different combinations. So I'm going to tell you about one study with one type of mouse. This is not generalizable across the broad spectrum, but it is a really fascinating study that came out about 8 weeks ago that I want to share with you. So I'm going to start by saying a number of individuals with autism have digestive issues, have chronic digestive issues, inflammatory bowel, irritable bowel, inflammatory bowel disease, Crohn's disease. And so there have been some studies looking to see, is there a difference in the gut population, the microbial population, in children with autism and in children without. And there are small studies, and many of them have found that yes, there is a difference in gut population, but there's a really important caveat here. Many kids with autism have antibiotics, often because of other things associated with autism, and many of them are on special diets because parents have tried to change the diet to try to cut out things that might influence certain behaviors. So you would expect that if you're on antibiotics a lot or if you have a very different diet, your gut microbiome is going to be different. So I think the finding that there are different microbiomes is somewhat suspect, and it might be that there are different microbiomes because of factors that have nothing at all to do with the symptoms of autism, but more because of the way people or parents respond to try to modulate those symptoms. Still, there are some mouse studies, and there is a specific model of mouse called the maternal immune activation mouse. And this mouse was developed because some cases of autism, again, not all, a small number of cases of autism seem to be linked back to a maternal infection when the woman was pregnant. Now, that's a small number. That does not mean every child with autism had a mother who had an infection, nor does it mean every woman who has an infection while she's pregnant will have a child with autism. Hear me there. Be careful if you talk about that. Make sure you say that appropriately. But there's a small number of women that had infections when they were pregnant, and they have a higher incidence of children with autism. So the MIA mouse model, they infect the mouse with something that is not infectious at all, but it mimics an infection. So they inject the mouse when the mother mouse is pregnant. And the mouse pups have a higher incidence of autism-like symptoms. They have repetitive behaviors. They bury marbles in the sand over and over and over. There's excessive grooming. They're antisocial. They don't interact with other mice. They have vocalization differences. They sound different than other mice. And they have higher levels of anxiety. So this is a model of a type of autism, this MIA mouse model. These children of the mothers that have these autism-like symptoms have been found to have a different population of bacteria in their gut than the children, the exact same genetically identical mice of moms that did not receive the injection. And they were found to have something called leaky gut. And I don't know if you've heard leaky gut. It's something that comes up in our vocabulary more and more and more. Leaky gut is exactly what it sounds like. The cells in your small intestine, let's go to the next image. The cells in your small intestine are normally very tightly joined together. So here are all the different cells. And there are proteins called tight junction, gap junctions, that hold the cells together and don't allow anything to come in between them. And this is really important because this keeps the bacteria and the food that are inside your digestive system from passing through the cells of your gut and making their way into the bloodstream the way your nutrients do. So individuals that have leaky gut syndrome, or more appropriately, membrane permeability, over here on the left-hand side of this image, have spaces in between the cells. This is an example discussing specifically celiac disease, and it's showing gluten fragments, but bacteria pass through these junctions as well and make their way into the bloodstream. So in these mice, these MIA mice, the females are injected with something that stimulates an immune response. The pups show autism-like behavior, and they develop a leaky gut, which is also found in some of the children with autism. So the researchers in this particular study took these mice and gave them a probiotic, Bacteroidetes fragilis. It's a bacterium that's found in humans. So they gave this to the mice, and it was given to them through their water, and they found that it sealed the leaky gut. So it reduced the permeability of the cells. So now you don't have bacteria passing through the gut and into the bloodstream. And it also restored the population of bacteria so that now these mice have bacterial populations that look like the bacterial populations of the mice that aren't affected with the symptoms. The amazing piece about this is that this treatment with this probiotic reduces some of the behavior features associated with autism in these mice. This is a paper that came out in Cell in December, and it hit lots of stories. You can imagine it made the autism boards light up, and lots of families were asking about this. And it was on NPR, and it was in Time, and it was in lots of different storylines. So the anxiety of these mice, the communication style of these mice, and the repetitive behaviors all modulated and appeared more like the typical mice. Now, we're not going to spend a lot of time on these images, in part because I want to move us on to get to the rest of our stories. But essentially, for each of these images, the black bar represents the control mice, and the white bar represents the mice that are born to the mothers with the mock injection. So they have, you can see they spend less time in open fields. This is if they're in the box. They spend less time out in the open. It's a higher measure of anxiety. They have more marble burying. That's the white ones, which is in this figure labeled C. But the blue bars are those mice that were given the probiotic in their water, really from weaning. Right after they were weaned. And you can see that they spend more time in the open box, the open space in the box if you look at A, and in C there's less repetitive burying of the marbles. And then in this image, this is looking at their vocalizations and the ultrasonic, the style, the number of vocalizations, the frequency. The mice that had the probiotic from a very young age, much more similar to the mice that didn't have any of the autism-like symptoms to begin with. Now, social disinterest didn't change. So the avoiding, the not spending time with other mice, spending more time with inanimate objects as opposed to a neighboring mice, that doesn't change at all. Giving them the probiotic didn't make a difference. Okay, hear me. This is one study in mice that have a set of symptoms like autism that are not exactly autism from one specific way that that was developed. This cannot be extended to this cannot be extended to humans, period. This cannot be extended to every form of autism. So don't take this and say, you now need to do this. This is not a go and do this. Do not do this at home. Do not tell friends that you have children that may be somewhere on the autism spectrum disorder. You need to go find Bacteroidetes fragilis and you need to start putting it in their water. Please. There are enough parents that already are asking about if they can do this. We are nowhere near this. I'm sharing this with you because it is an interesting mind-gut microbiome connection. A really fascinating one that may someday open the door to new discoveries. It's amazing. It made scientists sit up and pay attention in ways that they hadn't before. But this is not ready for prime time. Deal? Deal. All right. Yes?
What is the definition of a probiotic?
What is the definition of a probiotic? A probiotic is a live bacteria that you take in either in food or in capsule form for the express purpose of hoping that it has some beneficial effect for you. Maybe to help your digestive system be more regular, maybe in some of these cases to reduce mood issues. We're going to spend the 4th week of this series talking about probiotics and antibiotics and prebiotics, which are food that helps bacteria survive or helps beneficial bacteria thrive. And we're going to talk about all of those. You can find lots and lots and lots of probiotics in the grocery store and drugstore. Whether they're useful or not is still very, very much in question. Yes? And then I'll come here.
Isn't it just as likely that the bacteria got into the bloodstream, so they went through those holes, went to the brain, and then did something local?
Isn't it just as likely that the bacteria got in the bloodstream, went through the gaps in the leaky digestive system, and then found a way to get in the bloodstream and travel to the brain? So, the blood-brain barrier right off the bat would be a challenge. It would be difficult for the bacteria. It's not impossible. It would be difficult for the bacteria to cross. As opposed to if the bacteria, the thinking is that this bacteria confers an anti-inflammatory response. And so it calms the immune system from attacking the digestive tract, and that backing off of the inflammation allows the immune system to heal. It's somewhat similar to individuals that are gluten intolerant, removing gluten, so they remove that thing that inflames their immune system and allows the immune system to heal back. But I can see why you would ask that question. That's a really good question. Let me come here, and then I'll come to Mark. And then I'll see if there's anything online, any from our online group.
My gastroenterologist loves yogurt. Loves, says you can't eat enough of that. If these things aren't harmful, why in the world wouldn't you give it to your children? Just in case.
So, if there's all kinds of probiotics in yogurt, and if these things aren't horrible, why wouldn't you go ahead and give it to your kids just in case? Let me give you a couple of pieces there. In some instances, the probiotics just wave at your digestive system as they pass right through. In some instances, the probiotics that you think you're taking may not necessarily be the probiotics that are actually present. So it isn't. Some groups do a great job regulating the quality of their probiotics, and other groups don't. And there are certain bacteria that you do not want to introduce into your digestive system. And as we learn more about these bacteria, I think we're going to find that it's quite possible that some probiotics carry out this function and some probiotics carry out very different functions. And you may not want this function in your system at all times. You may not want something that influences mood or that calms inflammation continually. I think we're going to get to the point where we're going to ultimately know for this set of symptoms or for this window of time, this is the probiotic you should take. And certainly if you have a probiotic that you're on that works for you, I am not going to say you should quit taking it. But right now it is very much the wild, wild west in some of the manufacturers that what you think you're taking may not be what you actually are taking. Mark?
That was my question right there.
Why not just take it just because, just to be on the safe side?
Downsides to these folks.
This is really so, are there downsides to taking probiotics? This is really early going. And so I think before we just start inhaling huge amounts of every probiotic I've listed here tonight, I think we need more work to get a sense of that. And the first thing we need to know is which probiotics stay in our system and which ones just travel on through. Good question. Let's see if I have any online questions. Nope. Okay. All right. Now I'm going to shift gears. We're going to spend the next 10 minutes, and we might spill over a little bit into next week, talking about cardiovascular disease. And we're going to talk about this in terms of what you eat and what your gut and the microbes in your gut do with it and where that then goes. I like this. I saw this in a talk that I watched online last week. The largest environmental exposure is what we eat. Not the things we breathe in, not the things that are that we rub on our skin or the things we're exposed to like ultraviolet light. Our largest environmental exposure is the food you bring in your body. And we bring in kilogram quantities of foreign objects. And then our gut largely is the one that has to filter through that and decide what to do with that. And our microbiome plays a role in that. Now, the bacteria that live in your digestive system take some of this food or some of these chemicals and use them to make energy for themselves or break them down. And in some cases, the things that they make from that, their byproducts, are biologically active, like the neurotransmitters we just talked about. So your microbiome is taking in nutrients, just like your gut is taking in nutrients, and the enzymes in your gut are taking in nutrients, and it is turning them into things. And some of those things it's passing along to neighboring cells, or some of those things it's actually passing into your bloodstream that then passes all over your body. Now, that looks kind of good, doesn't it? The consumption of red meat has been linked to increased risk of cardiovascular disease. There are some studies that are unclear as to whether the saturated fats and the cholesterol are the primary drivers or are important but not necessarily the key risk factors for cardiovascular disease. Now, before I get any further into this story, I'm going to tell you, if you go home and you look this up online, you're going to find a lot of people that are going to tell you red meat causes heart disease, and a lot of people that say that's not true and that's just put together by people who don't like red meat. And you're going to find people very strong on both sides of the argument. I'm going to present you the data, recognizing that it's one study. It's one really intriguing study, though. So there are animal studies that link atherosclerosis, that link cardiovascular disease, to the microbes in your gut, specifically the microbes that take in a nutrient called phosphatidylcholine. And you find phosphatidylcholine in eggs, in meat, in dairy products. And these microbes take the phosphatidylcholine and they turn it into something called trimethylamine, which is abbreviated TMA. So let me show you this image. So here's the phosphatidylcholine in your diet, eggs, meat, dairy. In your digestive system, your body, your microbes take the choline and they turn that into trimethylamine, TMA. The TMA travels to your liver, and in your liver it's converted to something called TMAO. I know we got alphabet soup going on here, but I want you to, TMAO is what your liver does when it gets the TMA from the microbes in your gut. Everybody tracking with me? Okay, so you eat choline, The microbes in your gut take that choline and turn it into TMA. Your liver turns that into TMAO. And TMAO is being identified in some studies as a big driver in increasing the risk for atherosclerosis. This was in animal studies. So a group of researchers said, can we try to find some way to replicate this or to confirm that this is true in humans? So they took a group of volunteers and they had them come in for 3 separate visits. On the first visit, they gave them 2 hard-boiled eggs, which is a fairly good source of phosphatidylcholine. They ate the eggs. They gave them a little bit of choline with an isotope that allowed them to track it. So it was a way to kind of follow how all the choline got metabolized, both from the egg and the little bit that was in the capsule that they ate. And then they waited and they measured their blood and they looked to see how much TMAO is produced. So the idea being, you eat the egg, the choline goes into your digestive system, your microbes convert it into TMA, that goes to your liver, and it gets converted to TMAO. So this is what they saw. So from the number of individuals that they studied, there's a baseline level of TMAO in the plasma, in the blood, and a certain number of hours after they've eaten, or a certain amount of time after they've eaten the egg, the TMAO level goes up. And then after a while, it drops back down. So then, after that visit, they gave the volunteers in this study a broad-spectrum antibiotic to wipe out a lot of the microbes in their gut. And after being on that antibiotic for a week, they came back in and they repeated the test. No TMAO being produced. You still consume the same amount of choline, but the inference from this would be that you've destroyed the bacteria that would produce the TMA, that would produce TMA, that then your liver would convert to TMAO. So then, the third part of the study, they waited for about a month to let the volunteers' gut bacteria kind of repopulate. We'll talk about this in week 4. It takes a long time for your gut, for parts of your gut to repopulate after you've had an antibiotic. And they repeated the test, and so in the third panel, it's at lower levels, but we now are producing TMAO again. The gut bacteria, presumably, are converting the choline to TMA, and so on and so on. So then, these same authors looked at 4,000 individuals who were coming in for diagnostic heart catheterization. And at the time of the catheterization, they took a sample of blood and they measured their TMAO level. And they sorted all 4,000 people by their TMAO level into 4 groups. So the lowest quartile, the lowest quarter had the smallest amount, the highest quartile had the highest amount. And then they followed these individuals over a 3-year window, and they looked to see what percentage of them had an adverse cardiac event. So a stroke, myocardial infarction, death from heart attack, those all qualify as pretty adverse effects. And you can see at 1 year, and definitely at 3 years, there's a pretty significant difference in the frequency, the percentage of individuals with very high TMAO having issues, having adverse cardiac effects. Again, this is one study. This is still correlation. This is not directly linked to causation, but it's an intriguing approach. So then another group of scientists said, well, you know, TMAO, or TMA, isn't only made from choline, from eating choline, it's also made if you ingest carnitine. And carnitine is found in lots of things, but we're going to talk specifically about carnitine in red meat. Carnitine is a really important player in your cells. Carnitine takes fatty acids in your cells and takes them into your mitochondria, which is your powerhouse. It's where your energy is produced for your cells. And it brings the fatty acids in so that they can be turned into energy. So if you don't have any carnitine, you're not able to convert and produce energy. So I am not telling you that carnitine, I am telling you that carnitine has a very important role. But the researchers said, well, what happens if we follow the same approach that we did with the eggs, and 3 visits, and there's an antibiotic in the middle, and we give them a steak, And so in the paper, the author actually says, we used a George Foreman grill. Now, there are some issues with that because the process of grilling also creates some chemical compounds but we're going to set that as a challenge to the model. We're going to set that aside for a moment. And so in this case, we're looking at two things. So in these images, the lines with the squares are looking at the amount of the TMAO that's being produced, and the lines with the circles are looking at the consumption of carnitine. So you eat the steak, there's a lot of carnitine, and over time that gets converted to TMA and then to TMAO. And so you can see there in this first panel, the first visit, we're producing TMAO. In the second visit after the antibiotics, we are not producing TMAO, even though we're breaking down carnitine. And in the third visit, a month later, when they now have a new digestive set of microbiomes, we're now producing TMAO again. So both in phosphatidylcholine and in carnitine, we seem to be producing TMAO. So then the researchers said, well, is there a difference between the levels of TMAO for individuals that are vegetarians and don't normally eat meat and individuals that are omnivores that periodically eat red meat? So they convinced an individual who's a vegan to have steak. I'm not sure how that conversation went. And they measured the TMAO level of this individual and of an individual who is an omnivore who routinely eats red meat. And this is a study of 2 people. Be very skeptical about studies of 2 people. They built on this study, but they're showing this as an example. And you can see in this one individual, we are not producing TMAO from the vegan, and we are producing it from the omnivore. So they then went on to look at a number of individuals who are vegan, vegetarian, and omnivores, and said, just in your normal blood, if you're fasting, what's your baseline level of TMAO? Do vegetarians and vegans have any TMAO? And the answer is yes, they do. It's at a slightly lower level. So this box plots again. You want the line that's in the center of the box as the average across 26 vegan vegetarians and 51 omnivores. So slightly larger numbers, but still not enormous. And then they said, let's take some vegan and vegetarian individuals and let's give them carnitine in a capsule, because let's not try to force them to eat steak. Let's just give them carnitine in a capsule and see if they have the microbiome that converts it to TMA and then gets converted to TMAO. And almost non-existent, very low levels of TMAO production in the vegan and vegetarian population compared to the omnivore. So based on that, I'm going to take 2 more minutes of your time, and those of you that are in our online audience, and then I promise I'll close this down. I want to take you to this next slide, and we'll pick this up next week. They went to studies in mice, and they divided a group of mice into 4 different populations. Mice that were fed a regular diet, mice that were fed a diet that was enriched in carnitine, mice that were fed a regular diet and were given a broad-spectrum antibiotic, and mice that were fed a diet rich in carnitine and given a broad-spectrum antibiotic. And the 2 questions they're asking is, if I feed mice a diet rich in carnitine, do I get more atherosclerotic plaques building up in their vessels? And if I give these mice antibiotics and I reduce their microbe population, does that change the frequency of atherosclerotic plaques? So this image here in the center, the red are the atherosclerotic plaques, and you're looking essentially to see how much red is in this. And there's a lot more red in the mice that were fed a carnitine-rich diet than the mice that weren't. So that's the figure at the upper right compared to the upper left. And then if you look at the mice that were given either regular diet or carnitine but given a broad spectrum, there's very little atherosclerotic plaques. These authors infer that the antibiotics wipe out the microbes that create from carnitine the trimethylamine, the TMA, that then is converted to the TMAO. And next week we're going to talk a little bit more about what's the potential mechanism behind this. But I need to make sure that you don't walk away thinking you need to get rid of carnitine in your diet, because other studies have shown that carnitine is actually very helpful for patients that have just had a heart attack. So this is one study looking at one mechanism, and it may be that carnitine is very helpful unless you have the specific bacteria in your gut that convert it to TMA. And then that leads to the TMAO and increases the risk. This is by no means a settled discussion. So I am not telling you not to have a good steak this week. I'm also not telling you not to have a good steak this week. Carnitine is in a lot of energy drinks. So be aware of that, but I'm not telling you you shouldn't have it. The data are really uncertain at this point. But what is fascinating is attempting to make these links between what we eat and how the microbes in our gut try to break that down and turn that into active products that then carry out totally different sets of features in our body that we aren't even until now aware of. So next week we'll finish up this story briefly, and then we're going to jump into a little bit of cancer, and then obesity and type 2 diabetes and digestive disorders. We have a lot of ground to cover next week. Have a good night, be home safely, and I'll see you next week.